Hi everyone, today we are going to talk about gluconeogenesis. So, glucose can be synthesized from non-carbohydrate precursors. The glucogenic pathway converts pyruvate into glucose. Non-carbohydrate precursors of glucose are first converted into pyruvate or enter the pathway at later intermediates such as oxaloacetate and dihydroxyacetone phosphate. So, it's the pathway of gluconeogenesis. The reactions and enzymes unique to glycogenesis are shown here in red, and the other reactions are common to glycolysis. The enzymes for gluconeogenesis are located in the cytoplasm, except for pyruvate carboxylase and glucose 6-phosphatase. The entry points for lactate, glycerol, and amino acids are shown here. Glycerol is a precursor of glucose, but animals cannot convert fatty acids into glucose for reasons that will be given later. Glycerol may enter either the gluconeogenic or the glycolytic pathway at dihydroxyacetone phosphate. The conversion of pyruvate into phosphoenolpyruvate begins with the formation of oxaloacetate. The first step in glyconeogenesis is the carboxylation of pyruvate to form oxalatate at the expense of a molecule of ATP, a reaction catalyzed by pyruvate carboxylase. The reaction occurs in the mitochondria. Pyruvate carboxylase requires bonding a covalently attached prostatic group, which serves as the carrier of activated carbon dioxide, CO2. The carboxylate group of bottom is linked to the amino group of a specific lysine residue by an amide bond. A subunit of pyruvate carboxylase, like the bottom are covalently attached to the BCCP, is a transporter carbon dioxide CO2 from the BC active site to the PC active site of an adjacent subunit. Oxaloacetate is shuttled into the cytoplasm and converted into phosphoenolpyruvate. So first, we're going to introduce the compatible cooperation. Oxalate used in the cytoplasm for glyconeogenesis is formed in the mitochondrial matrix by the carboxylation of pyruvate. Oxaloacetate leaves the mitochondria by a specific transporter system in the form of malate, which is reoxidized to oxaloacetate in the cytoplasm. Oxaloacetate must thus be transported to the cytoplasm to complete the synthesis of phosphoenolpyruvate. Oxaloacetate is spontaneously decarboxylated and the phosphorylated by phosphoenolpyruvate carboxykinase is short for PEPCK to generate phosphoenolpyruvate. The conversion of fructose 1,6-biphosphate into fructose 6-phosphate and acetophosphate is an irreversible step. On formation, phosphoenolpyruvate is metabolized by the enzymes of glycolysis, but in the reverse direction. These reactions are near equilibrium under intracellular conditions, so when conditions favor glyconeogenesis, the reverse reaction will take place until the next irreversible step is reached. Generation of glucose from glucose 6-phosphate. So we're going to introduce this. Several endoplasmic reticular like ER proteins play a role in the generation of glucose from glucose 6-phosphate. T1 transporters like glucose 6-phosphate into the lumen of the ER, whereas T2 and T3 transport the phosphate 2 and glucose respectively back into the cytoplasm. 6 high transfer potential phosphorus groups are spent in synthesizing glucose from pyruvate. The formation of glucose from pyruvate is genetically unfavorable unless it is coupled to reactions that are favorable. Compare the stoichiometry of gluconeogenesis with that of the reverse of glycolysis. Gluconeogenesis and glycolysis are reciprocally regulated. So the first important regulation site is the interconversion of fructose 6-phosphate and fructose 1,6-biphosphate. Reciprocal regulation of gluconeogenesis and glycolysis in the liver. So the level of the fructose 2,6-biphosphate is high in the fat state and low in the starvation. Another important control is the inhibition of pyruvate kinase biphosphorylation during starvation. The balance between glycolysis and gluconeogenesis in the liver is sensitive to blood glucose concentration. The domain structure of the bifunctional enzyme phosphatidylfructokinase 2. The kinase domain, shown in purple here, is fused to the phosphatase domain, shown in red. The kinase domain is a P-loop NTP hydrolysis domain, as indicated by the purple shading. The bar represents the amino acid sequence of the enzyme. Control of the synthesis and degradation of fructose 2,6-biphosphate. So we're going to introduce this. A low blood glucose level as cyanated by glucagon leads to the phosphorylation of the bifunctional enzyme and hence to a lower level of fructose 2,6-biphosphate, showing glycolysis. Insulin accelerates the formation of fructose 2,6-biphosphate by facilitating the dephosphorylation of the bifunction enzyme. Substrate cycles amplify metabolite signals and produce heat. So we're going to introduce this part. A pair of reactions such as the phosphorylation of fructose 6-phosphate to fructose 1,6-biphosphate and its hydrolysis back to fructose 6-phosphate is called a substrate cycle. So this ATP-driven cycle operates at two different rates. A small change in the rates of the two opposing reactions result in a large change in the net flux of product B. Lactate and alanine formed by contracting muscle are used by other organs. The Cori cycle we're going to introduce here. So lactate formed by active muscle is converted into glucose by the liver. The cycle shifts part of the metabolite burden of active muscle to the liver. So the simple P represents nucleoside triphosphate. So let's now see the pathway integration. The cooperation between glycolysis and glycogenesis. So we're going to introduce this. During sprint, glycolysis and glyconeogenesis are coordinated. in a tissue-specific fashion and to ensure that the energy needs of all cells are met. So in skeleton leg muscle, for example, glucose will be metabolized aerobotically to CO2 and water, or more likely during a sprint, so it's like to lactate. In cardiac muscle, the lactate can be converted into pyruvate and used as a fuel, along with glucose to power the heart beats to keep the sprinter's blood flowing. Gluconeogenesis is primary function of the liver will be taking place rapidly to ensure that enough glucose is present in the blood for skeletal and for cardiac muscle, as well as for other tissues. So glycogen, glycerol, and amino acids are other sources of energy that we will learn about in later chapters. Thank you for your listening. Bye-bye.